Okay, so we have looked at a lot of regulation mechanisms, and so phosphorylation, dephosphorylation, we have seen they activate either kinase or phosphatases, and which ultimately then will regulate the activity of glycogen processing enzymes, glucose processing enzymes, to sort of balance the glycolysis versus gluconeogenesis pathways. One mechanism we have not seen yet, which is how transcriptional activity will regulate the enzyme activity. And so we'll see an example where two enzymes in the gluconeogenesis pathway, how they are inhibited under fed condition by insulin. So FOXO1 is a transcription factor, and the full name is fork head box protein 1. And we'll see how FOXO1 regulates the activity of gluconeogenesis enzymes. So there's a receptor, and this receptor will be activated by insulin. So this is the active form of the receptor. And then this will trigger a protein kinase B pathway, or this is the PKB pathway, protein kinase B. We have seen so far protein kinase A, which are activated by glucagon. We are now seeing protein kinase B pathway, which is activated by insulin. FOXO1 is the transcription factor where insulin is secreted, and it will trigger the protein kinase B to make it phosphorylated. Once it is phosphorylated, it is degraded, meaning it doesn't do anything. Once the phosphorylation happens, it is destroyed, the enzyme is destroyed. However, when the insulin levels are low, okay, and then under those conditions, what happens is that the FOXO1 will be dephosphorylated by the phosphatase. So there's a phosphatase which will dephosphorylate it. So once the dephosphorylation happens, then the dephosphorylated form of FOXO1 will enter nucleus. So it will enter nucleus, and FOXO1 here will actually bind to DNA, okay, will bind to DNA near the gene cluster that triggers the synthesis of gluconeogenesis enzymes, for example, phosphoenol pyruvate carboxykinase or PEPCK and glucose 6-phosphatase. So what this will do is the FOXO1 binding will trigger the synthesis of mRNA, and the mRNA, we know that we make RNA from DNA, and from RNA we make proteins, right? So the basically all the protein synthesis signals are in the mRNA. So these mRNAs will then ultimately trigger the synthesis of gluconeogenesis enzymes, PEPCK and glucose 6-phosphatase. So gluconeogenesis will be upregulated once these are synthesized. Right? Basically what happened, the big picture is that insulin is present under fed condition, and it will phosphorylate FOXO1 by the protein kinase B pathway. Once that phosphorylation happens, it will be targeted for degradation. It is not going to be active. So obviously in the fed condition, we don't want gluconeogenesis to happen, right? So under fed conditions, the enzymes that promote gluconeogenesis will be downregulated. It makes total sense. Once the insulin level is dropped, then FOXO1 dephosphorylation will happen by phosphatase, and then the FOXO1 in the dephosphorylated form can enter the nucleus, and once it enters the nucleus, then it will bind to the DNA region, which will trigger the synthesis of mRNA, ultimately making PEPCK and G6Pase, which are gluconeogenesis enzymes, so that gluconeogenesis can be upregulated. So that's a transcriptional regulation by which the gluconeogenesis enzymes are upregulated. So we have looked at many different types of enzyme regulation. So there's a general theme which I want to get across. And so certain enzymes are activated when they are phosphorylated, and certain forms of enzymes are deactivated when they are phosphorylated. There are basically two different metabolic states. One is the starved state. So in the starved state, what we have seen, the phosphorylation will upregulate the activity of enzymes which will release glucose. and it will downregulate the activity of enzymes which utilize glucose to do something with it, so maybe glycolysis. So in the starved state, we have seen FBPS2 and glycogen phosphorylase are activated because they control processes which release glucose. Now in the starved state, the enzymes that are deactivated by phosphorylation will be those which consume glucose. For example, these enzymes we don't want to be active because they will utilize glucose to do something. So PFK2 will upregulate glycolysis, pyruvate kinase will upregulate glycolysis, glycogen synthase will upregulate glycogen synthesis, so basically storage of glucose. But under starved state, we don't want to store glucose. We want to give glucose to other tissues. That's why these enzymes down here are deactivated and these enzymes down here are activated. So now in the fed state, it is opposite. The enzymes that consume glucose to do something with it, either do glycolysis or store as glycogen, those will be dephosphorylated, and the dephosphorylation will upregulate their activity. On the other hand, the enzymes that release glucose, their dephosphorylation will inhibit their activity, right? So it just makes sense because in the fed state, we want the enzymes that utilize glucose to be active and the enzymes that release glucose to be inactive. So all of these enzymes are activated by dephosphorylation. So in these cases, dephosphorylation actually prohibits their activity or downregulates the activity, because in the fed state we don't want glycogen phosphorylase to be active, or we don't want the F26BP to be hydrolyzed, because in the fed state we want the PFK2 to be active, so that's why FBPS2 is inactive. So you see the big picture is that under fed state or in the starved state, the enzyme activity is differentially regulated depending on phosphorylation or dephosphorylation. In the starved state, phosphorylation will trigger or make the enzymes active, which will release glucose, and it will make enzymes inactive that utilize glucose. It is opposite in the fed state. In the fed state, the dephosphorylation will activate the enzymes that utilize glucose, and dephosphorylation will inactivate the enzymes that don't utilize glucose, rather they release glucose.